It's a pleasure to be here, and I'd like to thank the organizers for giving me the opportunity to present this work. So I have to have some pictures of some gigantic grasses. In the middle, we have a picture of switchgrass, which is poised to be an important biofuel, biomass feedstock. And as you heard before, however, switchgrass, its relatives, and really most grasses are very difficult experimental subjects. So we actually use this tiny little plant over to the right, Brachypodium, for a lot of our research. And that's because it's small. You can fit 100 times the number of plants into the same amount of greenhouse space. Has a fast generation time. It's self-compatible. You can efficiently cross it. John's lab has generated over 20,000 tDNA insertional knockout lines, and it has an expanding community, which is highlighted by the publications and the lines that have been requested. But it's a grass, and it has grass cell wall structure, development, and susceptibility to viruses and fungal pathogens. I'd like to note that this data from this project's already been used, from this CSP project has been used to find map and clone the first barley mosaic virus resistance gene. And that's despite years and years of work in barley to identify an equivalent gene. So we think that Brachypodium can really accelerate research in grass. It's a good candidate for the study of grass genome variation because it has a small genome, a number of other attributes. In particular, there's thousands of diverse inbred accessions. It's naturally inbreeding, and this is a huge resource. And those have been characterized to some extent, both genotypically and phenotypically. And so this study was to look in more depth in these natural inbred accessions. This is showing the actual plants that were sequenced in this study, actually a subset of them. And you'll note that they vary quite widely in their overall size, their stature, but also in a number of phenotypes that are more difficult to see, such as the flowering time, drought tolerance, disease resistance, digestibility of the biomass, seed shattering. This is just showing a subset, but there's 54 that have been sequenced in total. I'm going to talk primarily about the first 6. One phenotype that we're particularly interested in is tolerance to abiotic stress. So in the middle is Brachypodium accessions that were treated with a simulated drought. And you can see the accession on the left that has little or no wilting, whereas the accession on the right has severe wilting. So you can increase temperature, and that may get plants to grow faster, but if you don't give them water, they actually just die. So the idea here is that we can identify genes important for tolerance to drought, for example, and then engineer that tolerance into energy crops. And abiotic stress is particularly important for energy crops because we should grow them really on marginal land where we don't impact food crops. Also, it's expensive to irrigate, and water is an increasingly scarce resource. And the world is getting hotter and, more importantly, drier. This is showing the locations where a number of these lines were isolated from, and those are the red triangles. And the blue is a heat map of annual precipitation. So just to give you an idea of the environments that these lines come from. It's notable that there's a large field trial for drought tolerance, and it's called climate change. And actually, this Mediterranean region is a hotspot for increased drying conditions over the past 40 years. And that's expected to actually increase. So what does the data look like? We're sequencing these accessions fairly deeply, over 40x coverage. Most of the reads map. We generate a set of SNPs through multiple pipelines, and we generate a high confidence set through consensus. Looking at these SNPs, actually one accession in particular from Turkey has a large amount of private variation within that particular line. That's compared to the most geographically distant accession. And this made sense to us in the context of a previous diversity study that John had done using SSR markers, so low resolution within the genome, but a large number of accessions. And he could see that actually the late flowering accessions are down here. And they're very divergent from this group of early flowering accessions up above. So that made sense. But then when we used the CSP data, we actually saw that, that private diversity is localized within specific genome segments. So the orange box here is showing you a segment with a large amount of private variation for BD1-1. Whereas the adjacent region, the variation is much more well distributed, and BD30-1 then becomes the most divergent line in that segment. And that's the most divergent, the most geographically distant line. So this isn't just this particular region of the genome, it's actually throughout the genome. And if you correlate the frequency of your polymorphisms so within the genome, you actually get this anti-correlated pattern between BD1-1 and the other early flowering accessions, whereas the early flowering accessions are all positively correlated. The negative correlation is the green, and the red is the positive correlation. Why do we care? Well, the hypothesis here is that this pattern could be reflective of selection for some important flowering time genes. And if so, maybe we can use this data to identify those genes. So this is a collaboration with the Amasino lab. They're phenotyping the sequence lines for flowering and vernalization, also with the Mitchell Olds lab for doing some additional population genetics. I'd like to go to the second part of my talk, which is integrating quantitative RNA-Seq in with the CSP data. So the CSP data was looking at genomic variation, where are your SNPs, where are your large indels. But the younger lab, Tom and Dave, generated an RNA-Seq data set that we then tried to marry with the CSP data. So there's two samples per session, one well-watered and one water-deprived treatment. And they saw some very interesting patterns for specific genes. I don't really have time to give you much detail on that. Just note that there's 300 genes down here that they identified as having a genotype-dependent treatment response. This is actually trying to marry these different data sets. So this is using the CSP data to identify large deletions. So this is an area of about 90 kb. You can see that for 3 of the accessions, there's a large deletion that does not span this window, whereas 3 accessions have a deletion that spans the window. This is using paired end read information. If you look at the coverage of the genome, again, there's 3 accessions that have read coverage that extends in through this window, but 3 that don't. The reference, BD21, obviously has coverage throughout this area. If you look at the expression, there's 3 lines that have expression, 3 divergent lines that have expression as well as the reference, and 3 that don't. So the idea here is that you're knocking out genes. And these aren't just any genes. They're actually putative disease resistance genes in this case. There's a cluster of them right there over the deletion. So the idea here is that this deletion segregating in these Brachypodium populations, and maybe it's related to the susceptibility to a particular pathogen. That's just a wild hypothesis. If we look genome-wide and try to marry the deletion, gene expression data, we can see a really good concordance. If we just use raw data, we segment out regions that have that lack the start of new alignments or lack read coverage. So we can get 97, 86% concordance. I'd like to bring up another example, which is this part 3 of my talk is designing accession-tailored genomes. And I think it's important. I'm going to bring up an example, gene duplication, to highlight that. So again, marrying the genomic data and the RNA-Seq data, in the RNA-Seq data set we saw for this particular gene, there's 2 accessions that have really high expression in both of the data points, COS3 and BD1-1. And without additional data, you wouldn't really know what's going on. But with the genomic data, you can see double the read coverage in this area. So the idea is that there's a duplicate copy of this gene in some of the accessions, but not in others. For examples like this, these are really the interesting examples to me because why do we care? Well, if you overexpress the homolog of this gene and Arabidopsis imparts resistance to drought and salt stress, which is right in the context of where this RNA-Seq was generated. And the hypothesis here is that this duplication may impart some selective advantage under stressful conditions. So there's lots of these copy number variants. And to the extent that we think that these accessions deserve their own personal genome. So we've put a lot of effort in to try to build accession-specific genomes for them, incorporating, one way to do that is to incorporate the high confidence polymorphisms that we have into the reference sequence. In this particular, just these 6 accessions, that corresponds to about 11.3 megabases of non-redundant sequence that's inserted or deleted from the reference. If we take the RNA-Seq data and then map that to the new genomes, you see an increased number of reads that are mapping, and also the genomic reads map better, also more of them mapping as proper pairs. We're releasing this data on Brachypodium, and so we've put some effort to put up tracks for the SNPs and also small indels, and we'll be putting more and more data up there. So I just direct your attention there. And I'd like to highlight just briefly one additional project, which is genomes and gene annotations for 2 new Brachypodium species, Stacyi and Hybridum. So these were originally thought to be distachyon, but they're not. And they were reclassified very recently by a collaborator. So Hybridum in particular, I'd highlight here, it's an allopolyploid. And if you take its chromosomes and you stain them with probes from Stacyi and distachyon, then you can nicely highlight 2 different subgenomes. So the idea is that this is an allopolyploidy event between these distachyon and Stacyi. We've been generating a lot of sequence data. Also, we've generated genetic maps for both Hybridum and Stacyi. And we're generating a lot of RNA-Seq data to aid annotation. So I'd just like to put an advertisement for that project. I tried to highlight people in my talk, but in particular, I'd like to take this moment just to thank the people from JGI, Wendy, Joel, Lynn, Carrie, Zhang, Dan, and Jeremy. And in particular, Wendy and Joel for their time and also for the opportunity to be a visitor at JGI. Also like to recognize a large number of collaborators that are collaborating on these projects, on also related projects, and funding from DOE and USDA. Thank you for your attention.
Thanks, Sean. Are there any questions for Sean? So you showed some cases where there's sequence in the reference, but none of the other accessions you looked at had that NB-ARC region. Is that surprising to you? Is it a problem with the reference? Is it a problem with the resequencing? Maybe
No,
they're not actually missing?
I don't think it's a problem, but we will see that in the next 54. Mm-hmm. So it'll be really interesting to see at what frequencies these different events occur. I mean, those particular genes were present in 3 of the accessions and not present in 3 others, but the A deletion was present in that area.
Right.
If I give you the whole story, it's way more complex, because if you overlay the RNA-Seq data, you can see in the window just adjacent, actually some of the genes are expressed.
So they're moved.
So they moved. It's a local deletion, but there's sequence there somewhere in the genome. So these kinds of events really confound doing RNA-Seq experiments in these different accessions if you just use a single reference. And that's why we think having accession-specific genomes is useful.
Thanks.